Science Cafe. And welcome to our Science Cafe tonight. And our Science Cafe is organized by the UMass Chan Postdoc Association at the UMass Chan Medical School here in Worcester, Massachusetts. Our event tonight is on the topic of structural biology, cryo-EM, and how to use structural biology to understand viral infection. Before we introduce our speakers, I want to quickly remind everyone that the views in this presentation and the speakers' presentations are solely of the presenters and not necessarily those of UMass Chan Medical School. Also, our Science Cafe are recorded and will be uploaded to our YouTube channel here. If you want to share the seminar with your friends and colleagues, you're welcome to do that. Also, please follow our social media channels listed in the chat box if you would like to stay up to date with our recent events. And also, if you have a question during the presentation and have questions to the presenters, please type it in the chat box, and we will ask you the questions at the end of each presentation. Our first presenter today is Semena Zaltik. And Semena obtained her PhD from University of Quebec in Montreal in Canada, and she focused on vaccination technologies against the influenza virus. During her PhD, she did an internship in the during the PhD internship, she did research with Edward Engelmann's lab at the University of Virginia. And she was introduced to cryo-EM during that time. And she currently works as a postdoc in the Nicholas Grigoriev lab in the University of Massachusetts Chan Medical School. Her research explores the cryo-EM tools to advance the understanding of viral infections. And she's mainly focused on unrevealing how rotavirus, an important pathogen that causes diarrhea, hijacks the host protein synthesis machinery to facilitate the viral replication. Ximena is a native in Buenos Aires of Argentina, but she grew up in Quebec, Canada. Outside the lab, Ximena is a sport enthusiast, and she have a passion of baking cakes. And Ximena, here is your stage. You can share your slides.
Ximena, I think you're on mute. Are you able to see the right mode right now?
No, we're seeing it in presenter slide.
Oh, okay, 1 minute then. Let me just change it. Now it should be okay, I think.
Yes.
Is it? Yes, all right. And let me just put the last laser on and pointer options. All right. Okay. All right. Good evening, everyone. First, I want to thank you for being here. I know it is a wonderful day today, very good weather. So thank you for being sitting in front of your computer. I also want to thank the organizing committee of Science Cafe for giving me the opportunity to present here, and also for organizing these events that are really great. I'm really excited to be doing some outreach, and also very scared because that is not what I normally do. So I hope I will be able to walk you through how we can use cryo-electron microscopy to look at virus infection. So first thing first, what is structural biology? So for those that are not in the field, structural biology mainly focus into looking at how biological molecules fall in the 3D space. And here we mostly talk about protein, RNA, DNA, membrane, and sugars. And proteins are a very good example because they need to have the appropriate 3D folding in order to be able to function. So in order to be able to have the activity, and hemoglobin is a good example. Hemoglobin is a protein that actually carries the oxygen around your body, and it's a protein that you are seeing here now, and you can see here in this animation the oxygen binding into the hemoglobin. And these animation and the models are made out of information that we are able to get thanks to structural biology. And here again, you can actually see the oxygen bound into the hemoglobin. One thing I want to highlight here again is that the structure is key for understanding the function. However, it has been far from straightforward to be able to get those structures. So it used to be pretty hard and takes a lot of time to be able to get one of those structures. Oops, I cannot switch. Oh, all right. And so Cryo-Electron microscopy has made things a little bit easier. Often called cryo-EM. Cryo-EM stands for cryogenic electron microscopy. And cryogenic is because it's done in very, very cold conditions. So I had a question for you guys. I wanted to know if you have an idea of what are the conditions. So how cold is cryo-EM? And there will be a pool appearing, yeah, over there. So if you want to guess, you know, okay. Actually, I think, I don't know if I can share results. Yeah, you can see it over here, but most of the people say minus 195, which is actually the right answer. So let me see if I can, all right. Cryo-electron microscopy use samples that are at very, very cold temperatures. So we are freezing our sample at around minus 80, minus 185, and cooling our microscopes at minus 195, which is pretty cold. Actually, is the temperature of Uranus, which I think it's very cool. But now getting into something a little bit more serious, cryo-electron microscopy actually has been around for about 2 decades. And initially, when people started doing electron microscopy, they were called the blobologists. And that was because they were not able to get any high resolution structures. So their reconstructions were looking like potatoes, basically. Over the last decades, that has improved a lot, actually advances into computer science. So having more powerful computers, having better software that are able to get a better corrections from the images we get from the scopes, and having better cameras have allowed us to be much more better at doing reconstruction. And right now we are able to get high resolution models fairly fast. And actually all those advances happened close to 2015, which actually led to something that is often called in the structural biology field as resolution revolution. So I think at that point, the gates were open for structural biologists to be able to get fairly easy or much more easier structures. And also that is why the 2017 Nobel Prize in chemistry was actually awarded to 3 of the pioneers of cryo-electron microscopy. So now, how does cryo-electron microscopy work? So in a traditional way, how we prepare sample is that we start with a purified sample that can be, for example, a protein. So here we're talking about only one compound that we are adding into a cryo-EM support that we normally call grid. Then we are blotting the sample and plunge freezing it. So here the freezing is happening very, very fast. Again, here we use around minus 185 degrees Celsius as temperature. And what we want to do by freezing that fast is to avoid the formation of crystals that can damage our 3D structures. And then we are bringing the sample into the cryo-electron microscopes and taking movies and aligning those movies in order to get images that then are used to pick particles. So basically here what we understand is that we are gonna select little areas that contain only our molecule of interest. And then we can use those small images in order to classify and align them. So some sort of averaging, and then get a 3D reconstruction. And in the last years, we have became pretty good at it. And so we are faster and better. And actually, you can get a structure of a protein today in about less than a week. Of course, this is dependent on the protein, but once you get your sample, it can go as fast as a week. And so from there, we have been wondering how we can do better, right? How can we understand better how proteins fall and how protein interact in more complex situations, especially inside cells, right? Where things are quite different than inside a tube. And so for doing that, many or some methods have been developed. Again, this happened in the last decade, and one of those methods is the cryofib milling. And here, what we are doing actually is that we are adding cells into our cryo-EM support, or growing those cells into those supports. And then we are coming and cutting thin slices of those cells. So basically, this ion beam microscope is able to remove the material above and below of a very thin section of the cell. And that basically open windows to look inside a cell. Those samples are then added into a cryo-electron microscope, and then we can get images. And here you can see a little bit how one of those images look like. In fact, here you're looking at a rotavirus, and here a rotavirus inside a vesicle. If we compare that to a purified sample, I hope you can realize that it's quite different. And yeah, so this process of preparing the sample is a little bit more complex and than what I'm explaining now. So if you want to get more information about how this is happening, and it's pretty impressive, I will suggest you to use this QR code to look into a video. I don't have time today to really walk you through, but I think that video explained it pretty well. One of the main issues that we do have with cellular sample is that we have many components in our images, right? So we have many different proteins, and sometimes we don't know the nature of them. The big issue here is then understanding what is what, right? And in order to try to address that problem, our lab, the Grigoriev lab, has been working on developing a new method that is called 2D template matching that helps to identify molecules in a cellular context. How this works, so we start with a cell. We take those 2D images that I was previously describing, and from there, we are using templates. I cannot search. Oh, all right. So from there, we're using templates and to try to identify those molecules. Basically, what we do is that we take models that are already available in a different database, and we create a template. So we create a 3D out of it. And then from there, we can do 2D projections in different orientations, right? Because the difference of the 3D and the 2D, in order to describe this 3D, we need to have many, many orientations. So we create those 2D projections, and then we're able to do cross correlations in order to compare the images with our 2D projections. And depending in the correlation, we are able to detect targets. And it was by using this technique that my colleague, Rowan Lucas, she was able to look at ribosomes and identify a small molecule that was bound to this ribosome, named tRNA. So here the ribosomes are the molecules that usually produce the proteins. She was also able to observe a drug bound to the ribosome, and that was because she did treat the cells with that drug. One of the things I also want to point out here is that she was able to reach pretty high resolution. So here we're talking about 3 angstroms in some areas. And for structural biologists, talking about angstroms seems something very common. However, I think it's not everyone is used to work with angstroms. So I wanted to ask you guys if you know what is an angstrom, right? So there should be a little question appearing soon. It might take a couple of minutes. That will give you time to think of it. Oh, okay. There. Oh, that's not the right one. Oh, there you go. So how small is an angstrom? All right. Okay. So again, you guys got it right. It's the size of an atom. Basically, an angstrom is pretty small. It's actually 10 to the minus 10. So about the size of an atom, which yes. All right. Let me just close that. Great. Okay, so now changing gears, what can we do with those techniques that I was talking to you about? We can study, for example, pathogens. So in my case, I am looking into rotavirus, which is an important pathogen. Actually, rotavirus still the leading pathogen of diarrhea-associated mortality worldwide. It mostly affects children under 5 years old and elders individuals, and there is a vaccine that has been adopted in many vaccination programs worldwide. However, there is no treatment or drug available. And so here you can see some images of rotavirus. Those are cryo-EM images of purified virus. Cool. So now, how does rotavirus get into your cells and get you infected, right? So the first thing that happens is the virus interact with your cells. Then it gets into vesicles, and later the virus break those vesicles in order to release a part of the capsid inside the cytosol, so into the inner part of the cell. So in the cytosol, rotavirus will release its genomics material, and then the genomic material will be used as a scaffold in order to produce viral proteins. Then those viral proteins will be shipped into a organella that is called a viroplasm that is specific to virus. So you won't see that in healthy cells. And here, the proteins are put together in order to form the virions. Here we're talking about immature virions, and those virions will then be maturated and then will be able to bug out of the cell. Here in your right, you can see an image of rotavirus going from the viroplasm into the ER. All right. The part of the infection I am mostly interested on is the translation. That is when the RNA, in this case, is translated into protein. So why I am interested into that part of the infection is because viruses don't have ribosomes. So they need to be able to use the ribosomes from the host. And again, the ribosomes here are the molecules that are able to produce protein. And so they take the ribosomes and they use them to produce their own viral proteins. However, in healthy cells, the ribosomes are very well controlled, and actually the protein synthesis is a mechanism that is tightly regulated. And so the virus need to be a little bit sneaky and have a couple of manipulations to do in order to be able to get proteins. And so for those that are not into the translation field, I just wanted to say that in mammalians, the ribosomes are often called the ADS and are formed out of a large subunit that we see here in blue, that is the 60S, and a small subunit that is here in yellow and called the 40S. So as I was saying, the making proteins is very highly regulated in cells and requires a couple steps. And the first step is what we call the initiation, which is basically bringing together the 60S and the 40S. So once those two subunits are together and they find the mRNA, they can start the elongation, so start making protein, and then this process is terminated. Again here, this is very well regulated, especially the initiation is a step that is pretty regulated by the cell. And so actually, here it seems quite simple, but initiation is a very complex step. It's a very complex mechanism, I'm sorry. So if we think of how the initiation happened, there's many things that need to really be there in order to start translating protein. And so initially we're gonna have the 40S, so the small subunit, and to that some initiation factors, so some small proteins need to be added, as well as some RNA. And from there, again, some other initiation factors need to be added, as well as the mRNA. And once we have the mRNA and a bunch of those initiation factors, then we have a conformational change of that complex that here is actually called the 48S. And so what is important here to remember is that once we have the mRNA, we're gonna be calling it 48S. And so once this happened, this last step where we change conformation, then we are ready to have the 60S, the large subunit, joining and having a ribosome that will be translating protein. Here there is actually many things happening, but the message that I want you to remember is that in the initiation we have many players, many complex, and which makes this process quite complex. Again, complex and complex. All right. And so rotavirus has figured its way to really manipulate the initiation. And we do know that there is a viral protein that is involved into the initiation. So to start these proteins translation. And we also know a little bit about how the viral mRNA are made of. However, we do not have a full story. We don't understand very well how all that comes together. And it is still unclear how this viral protein that has been reported as affecting the initiation really make having a lot of viral protein. Actually, something that is important to know here is that in rotavirus infection, we stop producing host proteins and we mainly do viral proteins. So we're still not understanding how this is happening, and so I wanted to understand better how rotavirus initiate the protein synthesis. And to do that, what I'm doing is that I'm growing cells that I am then infecting with rotavirus. And I am creating little pores into those cells, which then allow us to take the cytosol of the cells and put it all into the Cryo-Electron microscopy supports. So we're getting a lot of cellular material, most of it actually. And we're doing that very fast, so in about 10 minutes. And so we think we're able to conserve the native conditions by doing this protocol. And you can see here an image of our preparation. So if you look carefully, you can see here, those are membranes. And if you look over here, this is a rotavirus particle. And we have here, and here more rotavirus. All those rotaviruses that are in a vesicle are immature virus. So they are not infectious. They have been made by the cell. And we have some lumps here that are probably protein and might be ribosomes. So obviously here the problem is to identify what is what. And so I have been using this technique to the template matching that we have developed in our lab in order to identify the small subunit of the ribosome. And so what I do is I use a template that is available on the protein database. And I have been doing a little bit of a Frankenstein of that template because I'm removing a piece of it. So I'm removing the head of the 40S. The 40S has a head and a body. That is how we normally identify it. And so I remove the head. And when I look with only the body of the 40S into my images, I can locate those molecules. And from there, I can take those information and make reconstructions. And you can see here, one of my reconstruction, that is the 40S. And I hope you can appreciate that here we are also seeing the head of the 40S. So by only looking with the body, we can recover the whole 40S. We were also able to recover other initiation complex when looking with the 40S body. For example, we were able to recover the 40S that we can identify here because of the presence of the initiation factor 3 that is over here. And so, great, that is, we have a method here where we were able to look in a native condition, all those complexes that are affected by the viral infection. And so now I'm going to be showing you a little bit of my data. And so here, this is one of my reconstruction. This is the 48S initiation complex from rotavirus infected sample. Here, the resolution is pretty low, and that is because it's preliminary data. I'm working on it, and I am hoping to get a better resolution very soon. So if you look here at the electron density map, I hope you can see the head of the 40S as well as the body. We can also identify some of those initiation factors I was talking about. But one of the things I want to point out is this density that is over here. This density is only present in rotavirus infection, and it's actually the initiation factor 5B. The initiation factor 5B is only present in the ADS complex in healthy cells. So here, this is something special to rotavirus infection. And we are now trying to understand why the initiation factor 5B is found there. What is its role? And so we basically think that it does help the virus to get the initiation going. One of the other things I wanted to show you is this map, which is also a 48S initiation complex, where if you look in the density that is over here, this is in between the body of the 40S as well as the initiation factor 3B. So this density that I often call a weird density, we don't know what it is, but what we do know is that it's only observed in rotavirus infected sample. So far, we haven't been able to tell if that is protein or RNA, but we suspect that is helping the virus to initiate the translation as well. So now with all those little informations we have, we're trying to put all together into a mechanism and try to describe better how rotavirus work. And of course, one of the things we need here is to get higher resolution in order to be able to tell a little bit better what is the role of especially that very weird density present. So overall, structural biology is essential for understanding how molecules work and how they interact. And structural biology does benefit from cryo-electron microscopy, especially in purified sample. Finally, we are now moving to in situ, which means inside cells, and these offer unique opportunities to better understand how biomolecules work, how they function in a native environment, and how they interact. And hopefully these will help us to understand better host pathogen interactions and eventually to develop new therapies. And with that, I want to thank my supervisor as well as all the lab members. And I want to thank you for listening to my presentation. And if you have any questions or suggestions, please reach out to me. Thank you.
Thank you, Zamina, for sharing your work with us and giving us a tour that how cryo-EM came into existence, and you described the workflow also. That was quite informative. So we have a couple of questions here in the chat box. In an uninfected cells, can you also catch any 48S complex with ELF5B other than in ATS?
In uninfected cells, no, we are not seeing the initiation factor 5B in healthy cells.
Okay, so we have another question. What kind of cells do you use for your experiment, and do you anticipate getting different results depending on the cell type? And or do you use, for example, primary intestinal epithelial cells or cell lines?
Yeah, this is a very good question. So we are now using BSC1 cells, which are not the primary cells that are used that rotavirus infect. But these cells are used a lot as a model in the literature, and so that's why we're using them. We could expect different result if we were using, for example, primary intestinal cells. And that will be interesting to look at that and to compare, for sure.
Okay. So as you are looking for the higher resolution, what tips do you keep in mind for sample preparation?
Sorry?
For the specimen preparation, if you are going for the high resolution, what you will look
Yes.
for?
In terms of sample preparation, there is not much we do for the sample preparation other than trying to have thin ice. For sure, the ice thickness is limited by the material we are adding it to our grids, which is we're adding a lot of material, right? Because we have all the components of the cell, or the components, or a lot of the components of the cells going into our grid. But that's basically it in terms of high resolution. What we are pushing for now is how we can get better alignments, and how we can get a higher amount of particle per class.
Okay. Yes, so we have another question. For the binding of ELF5B in infected cells, is there possibility to develop new therapeutic drugs?
Yes. So the initiation factor 5B could be an interesting target for developing drugs. So it is not that surprising to see initiation factor 5B, because it has been seen for other virus, or for at least one other virus, and it seems to play a role. However, that virus is very, very different to rotavirus in terms of the genomic material. So this was not expected, but yes, it can be used.
Thank you. I think we don't have any more questions. And if anyone has questions, please reach out to Zemina. Thank you very much, Zemina, for sharing your
Thank
work.
you.
Yeah. Over to you, Oscar.
Yeah. Savannah, can you stop your sharing?
Yes. Stop sharing. All right.
Okay, our next speaker is Judy Huang. And Judy grew up between Beijing, China, and various cities in Canada. And she attended McGill University, also in Montreal, Quebec, in Canada. And she obtained her Bachelor of Science in Microbiology and Immunology. During her undergraduate research internship, she realized her love for studying viruses, which led her to pursue graduate studies at the University of Massachusetts Chan Medical School. Currently, Judy is a fifth-year PhD candidate in the lab of Dr. Celia Schaefer, and her research focuses on using cryo-ET to better elucidating how an important human pathogen, influenza virus, binds its cognate receptor on host cell surface to initiate viral infection. And Judy?
Hi, thank you so much for the wonderful introduction. I'm going to share my screen. I think it's been shared. Going to send it into, are folks able to see it okay? Okay, perfect. Thank you so much for inviting me here to share a little bit on how we can use cryo-electron tomography actually to visualize viral infections. And thank you so much for to Ximena for having such a wonderful presentation, which I believe helped share some primers about structural biology and cryo-EM that will be really useful to hold for this presentation as well. So, well, I'll share what cryo-ET actually is in the next couple of slides. One of the really important things I wanted to share with you all is that cryo-electron tomography really enables imaging across a wide variety of scales. This is a diagram or a schematic that shows various imaging techniques from light microscopy to NMR, and as you can see, each of them can image different specimen at different sizes. In particular, the technique that I'm really interested in, cryo-electron tomography, can capture images from microns all the way down to sub-nanometer. And how does it do that? Well, maybe you'll find out within the scope of this talk. So what exactly is cryo-electron tomography? It is a sub-method of cryo-EM, cryo, of course, here being cryogenic again, and as we learned earlier, it means that your sample is at a very low temperature, typically below 180 degrees Celsius. Electron here really just means that the imaging source, or what we use to interact with our sample, and the radiation source is an electron beam. And tomography here, the etymology stems from two Greek words, tomos meaning slice or section, and grapho means to write. Essentially, taking those together, what you really want to do here with cryo-ET is to take your sample and look at it as a collection of slices. And this is how we're really able to capture a 3D view of a non-homogeneous specimen. Cryo-electron tomography is really well used for things like membranous viruses that are not completely uniform to whole prokaryotic and eukaryotic cells, and in more recent years, even up to tissue samples for whole organisms. When you have your sample, you first freeze the grid, as Simona explained earlier, and then what one can do is tilt the sample on the stage it is mounted on in the microscope and collect images at a variety of different orientations, oftentimes from minus 60 degrees all the way up to plus 60 degrees. And the images that you then take from all angles constitute a tilt series as you tilt your sample through this particular stage. And this tilt series can be projected into a 3D reconstruction of your sample, and henceforth is called a tomogram. And this is how we're really able to take a 3D view of a non-uniform sample by capturing views of it from all different angles and places. And lastly, the reason that we're able to get from tomograms with micron resolution to maps and reconstructions as sub-nanometer resolution is this idea of subtomogram averaging, where we can take the protein or component that we're interested in studying and align and average them all together to reach a consensus reconstruction at a slightly higher resolution. And this is just a video that showcases this particular process. And this shows a bacteria cell that is undergoing tilt series acquisition. It is on the grid, and then it is physically tilted over the variety of angles that I talked about, and this can then be aligned based on its common tilt axis to form this 3D volume that then we can visualize different proteins and cellular components in. That is one of the reasons that cryo-electron tomography often is referred to as a method that enables a window into a cell. And when we think of a cell, oftentimes we think of this simplified diagram where we have a cell, we have different components within the cytoplasm, such as, of course, the powerhouse of the cell, mitochondria, components like the nucleus, which holds our DNA, and different membranous components such as the endoplasmic reticulum, the Golgi apparatus, etc. But in reality, cryo-electron tomography has shown us that cells are very dynamic, being able to contain a wide variety of different components and very dense. The cytoplasm is a very crowded environment where proteins such as ribosomes and actin and cytoskeleton components are all piled upon each other, and you can also visualize components such as the nucleus, which is this very electron dense component, separated by a lipid bilayer that is the nucleolar envelope. So how can we then apply this method to visualize either viruses or infected cells that is undergoing and fighting off a viral infection? Cryo-electron tomography in the past 2 years have been able to reveal novel aspects of virus biology, from just the virion structures that discusses its components, what a virion is made of, to things like a virus's replication and life cycle and how it's able to hijack the cell to really allow us to see what a virus is doing in real time, as well as explore this idea of host-pathogen interplay and be able to really see how our cells or other organism cells are really interacting with viruses as it tries to fight it off, and the ways in which that a virus can then gain ability to fight back. So my thesis project really talks about influenza viruses, and they are envelope viruses that are extensively pleomorphic. Pleomorphic in this case means that they are extremely morphologically diverse. It holds 2 surface glycoproteins, hemagglutinin, which is the purple one depicted in this diagram, that is the major glycoprotein and helps influenza virions enter our cells. Neuraminidase, which is this blue one that is shown in less concentration. This also binds sialic acid, such as like hemagglutinin, but it cleaves it off to allow the virus to leave after it buzzed from the cell. It also contains its genome, which is RNA wrapped around proteins, and it constitutes the viral ribonucleoprotein complex. Influenza is really interesting because it has a very segmented genome. And lastly, it has a M1 protein layer that clings onto the underneath of the viral membrane, and even though this diagram doesn't depict it, it binds to both the VRMP genome as well as the cytoplasmic tails of the neuraminidase and hemagglutinin, Na, and HA proteins. And while this diagram doesn't necessarily show it, influenza viruses are very morphologically diverse, ranging from small and spherical to elongated to filamentous. And these are all virions that have the same genetic background. And this heterogeneity really stems from a very stochastic assembly process. And this is one of the reasons we study it using cryo-electron tomography, as conventional pure single particle cryo-electron microscopy typically uses protein assemblies that are more or less homogeneous, or virions that are non-membraned and have icosahedral symmetry. So in some of the work that I've been able to do during my PhD, I was able to identify the various components of the influenza virus when taken cryo-electron tomograms, as well as reconstruct some of the components. Now let me just play this video back again. Two of the proteins that are have been reconstructed using cryo-ET are hemagglutinin and neuraminidase, the matrix protein that cling to the underlayer of the membrane, and the viral ribonucleoprotein. So all in all, within the past decade or so, all of the influenza structural components have been investigated using cryo-electron tomography, and these really help us establish how a virion can look, as well as what components and what shape and form they tend to take. The next example I wanted to look at is how we can use cryo-electron tomography to investigate how SARS-CoV-2, which is a virus that many of us are intimately familiar with, can hijack the host cell in order to assemble new virions. And this is just one stage of the life cycle that has been investigated using cryo-electron tomography. So this is a slice through a cryo-electron tomogram of an infected A549 cell, and as you can see, coronaviruses are able to modify the cellular membrane to form these vesicles that then is able to use as sites for RNA replication as well as virion assembly. So over the course of these tomographic slices and the segmentation representations, you can see a membrane vesicle that is studded with the coronavirus spike protein. And after the viral ribonucleoprotein, which again is the genome, is incorporated in, we have this positive curvature forming within the membrane, and then the virion starts to bud out. And afterwards, it is able to pinch through, and then you have a newly assembled virion that is contained within these vesicles that can then go through the trafficking system to egress out of the cell. And the last example I wanted to talk about is concerning bacteriophages, which is actually the most abundant organism on Earth. This is just a micrograph of a infecting bacteriophage. In light green here, I have the phage. You have the phage head, and then you have the phage tails that bind onto the surface of a bacteria. And these virions are in the process of ejecting its genome into the bacteria right now. And what folks have been able to learn through cryo-electron tomography is certain bacteriophage actually form a nucleus-like shell. And this is actually a very interesting compartment, as it's never been seen in a phage before, but it's essentially able to exclude much of the actual host cytoplasm to form this proteinaceous shell made up of a phage protein that occludes protein transport. This is seen as a dedicated site for DNA to replicate, where then the DNA can be packaged into the green viral capsids that wait here, and afterwards they can then meet up with the phage tails and lyse the bacterium to escape. And this is a really wonderful example of host-pathogen interplay, as antiviral factors from the bacteria cannot enter this proteinaceous shell to then cleave the nucleic acid. Therefore, it protects the viral genome from being hurt by the bacteria's antiviral approaches. Hopefully, I've been able to convince you in this brief presentation that cryo-ET can really enable a window into the viral life cycle and enable discoveries in structure, replication, and immunity. But what are the next steps that we as a field can do then to improve upon our current understanding? And in the next couple slides, I'll just briefly discuss some future directions that the cryo-electron microscopy and tomography fields are engaging in, and hopefully in the next years can be applied onto viruses. In certain, this is from a publication published a few years ago, and this is a this is a bacteria cell, and researchers were able to obtain a 3.5 angstrom reconstruction of the bacterial ribosome. And one of the reasons that we're able to reach higher and higher resolutions is a advancement in the hardware, in the detectors and cameras, but also in software in terms for tilt series alignment, in terms for subtomogram averaging programs, and hopefully we can then leverage fewer terabytes of data to get better and better reconstructions. And this is just one of the ways that I think the field is trending towards. One of the other things that I think will be advanced in the next couple of years is to visualize the structures of dynamic macromolecular machinery in native cellular environments. This is just showing essentially a recent publication that describes a deep generative model that you can use to generate 3D density maps directly from subtomograms and can capture states that are diverse in conformation and composition. And just by playing this video through again, you can see the different sites of the ribosomes being occupied, and this is a really nice representation of how you can use tomography to really delineate the different states that proteins are undergoing in a particular cell. And this is actually, you can And one could possibly even apply this to a virus-infected cell to see how protein dynamics can be altered. And even though I'm talking about capturing more smaller, more granular conformations, one of the different approaches that's really been pushed in the field is this to moving towards investigating not just singular proteins for singular cells, but possibly tissues and even multicellular organisms. This is just an example that's been published recently on C. elegans, which is one of the most widely used models for genomics, for genetics research. And in a recent study, people were able to take a frozen C. elegans and take sections of it from different areas of its body and conduct cryo-electron tomography to really investigate in high resolution how the worm really changes in morphology as you go from a body part to body part. And this brings me to the end of my presentation. Thank you all for being here, and hopefully that was an enjoyable little primer into exploring viral infections using cryo-electron tomography.
Thank you, Judy, for sharing your work. And the basics of this complex technique you have explained with the video was quite interesting. So we have a couple of questions for you. First is, since influenza virus can do the cap snatching for translation, is there chances that cryo-ET can help to understand the cap snatch process?
Yeah, for sure. I think in both, cryo-ET and also in situ cryo-EM, like how Ximena was explaining, if we're able to take a cell, fit mill it thin enough, and obviously have it be like infected, then looking into how different components are engaged in cap snatching is possible. This might actually be a better case for in situ cryo-EM than for cryo-electron tomography, just because tomography tends to be a bit lower resolution. So if the factors involved in it are a little bit smaller, it might be difficult.
Okay, so the second question we have, what dictate the shape of the influenza virus particle? Does the 3D structure of its protein differ from viruses of different shapes?
So if it's all coming from the same genetic background, the different proteins don't really have any differences in terms of shape, conformation, etc. What really dictates the shape of influenza virus particles actually comes down to the M1 protein, which forms a oligomeric assembly underneath the membrane. And it's really interesting. The shape is actually dictated oftentimes by what organism it's infecting. If it's, for example, eggs or like very simple sort of organisms, it retains more of a spherical or slightly oval shape. If it's infecting an animal or even, polarizable cell types, it protrudes predominantly into filaments, and a lot of it is for immune evasion and also for better efficiency in infection. So there's specific amino acids, residues in M1 that really dictate the shape of your virion.
So what are the tools you use to identify different cell components?
Different cell
Components? What kind of tools you use?
components? Oh yeah. Yeah, so the components I guess when it started it was done almost completely manually. You will have people just manually annotating the shapes of proteins that they're able to identify. In more recent years, there's more semi-automated approaches where you can essentially train a machine learning algorithm, typically a neural network or the such, to learn the shape of the different components. Of course, this also it is also a lot of manual work because you really need to know what things look like to train a good network. But it eliminates a lot of the actual time to, go in there and pick all of your components.
So it seems that AI can have impact on the cryo-ET as well in the future.
Yes, in some ways, yeah. It's the one joke I always love to make is my artificial intelligence isn't very smart.
Okay, so we have another question. For C. elegans cryo-ET, is there any limitation in terms of size of the worm? Does the size shape of the worm affect the resolution?
Absolutely. So, for a cryo-EM or cryo-ET sample, the maximum sort of thickness that it can really endure before it goes into essentially dark density that isn't really resolvable is about 300 nanometers. So no matter the size of your organism of interest, you have to first freeze it in some ways. If it's thinner than like a couple microns or so, you could do plunge freezing. If it's thicker than a couple microns, you have to do high pressure freezing to make sure that your internal structures are vitreously frozen. So even if you have a worm that's say a millimeter long, right, you still have to freeze it completely and then cut into sections of it and then ablate away the material like what Ximena described in fit milling. And then in the end, you have this, say, for example, 200 nanometer slice that you're subjecting into the microscope. The thickness of your sample is one of the most important factors for resolution.
Okay. Thank you, Judy, for sharing your research with us. And if you have any question, please reach out to Judy. We have posted our survey forms for your feedback so that we can make it better in future. Thank you very much. And Oscar.
Yeah, thank you so much again to both of the speakers, and thank you all for joining. And again, finally, we very much appreciate everyone who joined for today's seminar to help us fill out the survey which has been posted in the chat box. This will help us to be better for the in the future. So thank you all for joining, and see you at our next event. Have a good night. Bye.
Thank you. Bye.